All right,
Looking
it's cool.
forward to
So I'm
it.
going to talk about some of the work that I did with John Rinzel and Yuri Bujaki at NYU, and then a little bit of follow-up work that I've been doing with Adrian Peyraj at McGill. Um, let's see. Okay, yeah. So basically, one of the earliest observations in neuroscience is that the brain does the most interesting stuff while you're asleep, right? So this goes back to the 1930s when Alfred Loomis would have his friends wear his new fancy EEG set when they took naps at his house. So he would have his, New York City friends come to his house and just take these naps and record whatever happened. And so what he noticed is that when they fell asleep, you start seeing these large fluctuations in the EEG signal, right? So these are larger than basically anything you see during wakefulness. And importantly, as they fell into what he called deeper and deeper sleep, the fluctuations got bigger and bigger. And so let's fast forward now 80 years. And so now we know that these large deflections from the EEG correspond to synchronized alternations in neural populations in the cortex between up and down states, right? So I'm showing here about 6 seconds of data recorded by Brendan Watson from the frontal cortex of naturally sleeping rats. And on top, you can see the LFP, or the intracranial equivalent of the EEG. And below, you can see the spiking activity of 62 simultaneously recorded neurons in the cortex. And notice that every time you have one of these large positive deflections of the LFP, you have silence in all of the neurons that we've recorded nearby. And we see this on the population level. Now, when we look at all of the neurons in the data set, when you align their spiking to the positive peak in the LFP, you see basically that all the neurons stop spiking. So these are our up and down states, which correspond to the slow waves in the LFP. But there's this big open question, which is what are the underlying mechanisms of these slow oscillations? And so this is kind of what we wanted to tackle during my PhD. And so fortunately, we didn't really have to start anywhere near from scratch. And that's because these synchronized up-down alternations are really the default mode of cortical tissue when it's offline. So they're not only seen during non-REM sleep, but you also see alternations between up and down states during quiet wakefulness and sensory cortex, as well as under anesthesia, and even in slice and cortical culture preparations. And so because of all this work, especially in the more in vitro preparations, um, there's been a lot of good modeling work, and it's all kind of come down to this canonical mechanism, which is that these alternations come from the interplay between recurrence and adaptation, as Mauricio also talked about earlier. And so basically what ends up happening is during the upstate, recurrent excitation maintains activity in the network, but also you have the buildup of adaptation currents, which eventually are strong enough to bring the network into a down state. The network then is able to recover from adaptation because no one is spiking. And then eventually the population is able to bring itself back into the upstate, um, either due to the loss of this adaptation current or the effect of noisy fluctuations, which set off this chain reaction again. And so what we wanted to do was to kind of build on this extensive modeling work, which has models a varying range of detail, and use it to model what to match to what we see in our data during natural sleep. And so what we decided to do was to kind of abstract this simple or this model into as simple a formulation as we could, which hopefully would allow us to study the behavior of the mechanism and how it relates to the effective parameters. And so we did this with an adapting Wilson-Cowan model. Um, Basically, the model represents the activity of a neural population. It has input from an external source, as well as recurrent connections in the network, and then subtractive current due to the effects of adaptation, where adaptation is turned on by the rate of our population and acts to self-inhibit the population, and it has some slower time constant tau a. We have sigmoid steady state functions for both the adaptation as well as the population input output function. And so what we find when we do an analysis of this model is that basically we have four distinct regimes of alternation dynamics, which I'm going to show you two ways. Once on the left with the phase plane, right? So trajectories, the dynamics of the model are represented as trajectories in the phase plane. Fixed points will be these solid circles. And I'm also going to go show you the time course on the left, where the dynamics of the population and the adaptation are shown over time. And so the first regime is an oscillatory regime in which we have a limit cycle around an unstable fixed point. And in the time course, basically the population alternates between active and inactive states, so regularly alternates between up and down states, during which the population goes back and forth between this right branch of the R-null-Klein and the left branch of the R-null-Klein. We also have a bistable regime in which we have two stable fixed points. But of course, these are metastable because we have noise in the system and we're close to these saddle points. So as long as the noise is big enough, we can be kicked back and forth between these two stable fixed points, and the trajectories or the time courses will show these alternations basically between an active and inactive state during which adaptation is active and inactive correspondingly. And then finally, we have two regimes which are excitable. So in each of these regimes, we have a single stable fixed point, but again, you're close to the saddle point. So noise can kick you over into the right or left branch of the R-null-Klein, before which you kind of cycle around and come back to the stable fixed point. And so in the time courses, the way these look is we have most of the time here in the excitable down regime, most of the time is spent down. But every once in a while, you could see that you have these transient excursions to an upstate, which are rather stereotyped. And so this is the trajectory in the phase plane going around the right branch of the R-null-Klein before it comes back to the stable fixed point. And the excitable up regime is basically the same thing, just the mirror image, where now the upstate is stable in the stable point. Adaptation is tonically active, but then every once in a while you can get kicked over into a downstate. Adaptation inactivates and eventually brings you back to the upstate. And the reason that we have these four different regimes is basically because the up and the downstate corresponding to the left and right branch of the R-null-Klein can either be metastable, right? So they can have a stable fixed point, which can give you noise induced excursions to the other branch, or they can be transient, meaning that there's no stable fixed point and you only need the effect of adaptation either turning on or turning off before you transition back to the other state. Okay, so the question we first had was, how do the parameters of the model determine which of these regimes you're in? And so we answered this by looking at a number of different bifurcation diagrams. I'm going to show you the bifurcation diagram with respect to drive and recurrence. And these are going to tell us where the transitions between the regimes happen in parameter space. So this is the IW bifurcation diagram. It has this configuration with the crossing of two cusps and a hop bifurcation. And so the result is you have this bistable domain at high recurrence, an oscillatory domain at kind of intermediate recurrence, and that three fixed point configuration of the excitable regimes with one stable fixed point kind of have this limited area in parameter space. Oh, and this is just to remind myself that if you look kind of back in the old bifurcation literature, this configuration is called a butterfly catastrophe, which is kind of a fun fact from history. But what does this actually teach us about up and down states? Well, first, that up and down alternations really require some sufficient amount of recurrent excitation. And they appear in this codimension 2 hop bifurcation, which we can actually in the model solve analytically, and we've mapped out where it occurs from where you can see here. But the crucial thing is that you need some sufficient level of recurrent excitation to maintain a self-maintained upstate in the population. And then the other thing is that if we look at the fixed points as a function of drive for different level of recurrence. Basically, this is the effective input-output curve of the population, right? So how does the steady state rate depend on the level of drive? And this can basically take two different configurations. It can either be oscillatory centered or bistable centered. And we have a transition here where the cusps cross, which again, we can solve analytically. And it comes at some intermediate level of recurrence where you transition between having oscillations to having bistability. And then so the final thing is that despite the fact that we had this, we only have the excitable regimes in this tiny area of parameter space, when we actually run the simulations, we find that the excitable regimes extend pretty far out into parameter space away from the butterfly. And so what I'm showing here is the results now of many simulations of the model and the mean durations of both the up and the down states, where red indicates longer ups, blue indicates longer downs. And so you can see is that as we increase the level of drive, the up states get longer and longer. And crucially, we're able to detect up and down states, so bimodality in the rate, in pretty wide range of parameter space. And so outside the butterfly, what you see is that we have long up states on the right, long down states on the left, and more importantly, that the coefficient of variation of the upstate durations is very long over on the right, while the coefficient of variation of the downstates is very short. And we have the flip side on the left side of the butterfly, where the down CVs are very high, so the downs are regular and long, and the ups are short and regular. And so this is basically happening because what you have next to our bistable and oscillatory domains in the input-output diagram is a metastable configuration where even though you only have a single stable fixed point, because you're close to the either this hop or saddle node bifurcation, you're able to get these single excursions out to the opposite state. Which is a form of metastability due to being close to this bifurcation. And importantly, what this means is that we can actually use the up-down duration statistics as a signature for which dynamic regime we're in. So notice here that in the oscillatory regime, your up and down state durations are all short and regular, while in the bistable regime, they're both long and irregular. And in the excitable up and down regime, one of your states, so in the case of the excitable down regime, your down state is longer and very irregular, while your up states are very short and regular. And so that basically, if we're to look at the up and down state durations, we can map that back to the model and say, where in the model do we see up and down state durations that match what we see in the data? Okay, so what do we actually see when we look at the data from our naturally sleeping rats? So here's the 6 seconds of data that I showed you before, and the thing that should immediately jump out to you is that the down states are, as a rule, very short compared to the up states and all about the same duration. And this should remind you immediately of one of the excitable regimes that I showed you before. And when we quantify this over the data set, we see that that this basically holds over all the recordings, right? So the up states are long, they all have a CV of about 1, while the down states are all short and have a CV of around 0.3. And so when we now go back and directly match the duration distributions to the data, so this plot shows the similarity or the KS distance between the upstate and downstate durations in the model and the data, we see that we have this band of high similarity between the model and the data in this excitable up regime, close to the onset of bistability or oscillatory dynamics. Okay, so you might remember now that I mentioned something about deeper sleep in the human EEG with more and larger slow waves as sleep gets deeper. And so we actually have something similar in the rodent, which we can quantify by power in the delta band, where deeper sleep has higher delta power. So the question we had was, how does this depth of sleep match with the model? And so to connect with the model, we again looked at the up and down state durations now as a function of delta power. And so notice that so this shows the upstate duration distributions as a function of delta power. And notice that while the downstate durations always stay about the same, regardless of delta power, what ends up changing is the upstate durations get shorter and shorter and slightly more regular. And so when we play the same game that we played before, basically matching these distributions from the data to what we see in the model, we see that as sleep gets deeper and deeper in the rodent, the best fitting parameter domain in the model gets closer and closer to the Hopf and saddle node bifurcations. And in some cases, so this would be some recordings that we have, it actually enters the oscillatory regime. So in these cases, the deepest sleep is actually an oscillation. Okay, so what have we learned so far about these excitable dynamics? First, that up and down alternations come in a few different flavors or different regimes based on the stability or transience of our up and down states. Second, that the neocortical slow oscillation is actually not usually an oscillation, but it reflects an excitable regime with a metastable upstate and a transient downstate. And so this means that the sleeping brain uses kind of this cool combination of noise and metastable dynamics to produce slow waves. And what I didn't show you is that actually we can fit the hippocampal data with this model too. So the hippocampal sharp waves are actually also well fit, except in this complementary excitable regime where the downstate is stable, but noise can kick you into transient upstates or sharp waves. And so for more, you can see this paper in Nature Communications with Yuri Buzhaki and John Rinzel. But I want to step back a little bit and point out that if we actually want to understand the mechanisms of slow waves, there's kind of this glaring open question, which is what's adaptation, right? We have a good sense of what recurrence is. Neurons are connected to each other, and that provides this positive feedback. But we've modeled this adaptation pretty abstractly, right? Basically, it can be anything that's slowly turned on by neural activity and acts to reduce neural activity. And this has been modeled and also shown experimentally. There are a number of different ways that neurons do this, right? There are things like voltage-dependent potassium currents, sodium-dependent potassium currents, short-term synaptic depression could even act like this, as well as GABA B. And our model can't really distinguish these in the sense that all of these different sources of slow negative feedback would have something like the excitable up regime that we see in the data. But I kind of thought at this point that this is where this project ends, right? We've done our job. We've laid out at least a general theory of the dynamics and its possibilities, and now we can kind of pass it off to experimentalists, and they can go figure out which of these sources is actually adaptation that's producing the slow waves. But then I started my postdoc with this guy, Adrian Peyrache.
5 minutes.
And he's awesome, perfect, thanks. And he says something to me around my first week in lab, which is that he has a student with some cool results that they can't really explain, and he was hoping that my model might be able to help. And so what he was talking about is that his student Dhruv was looking at some data collected by Adrian, in which they inserted a linear probe into the head direction cortex and were able to record the whole length of this cortical region from the dorsal to the ventral area. And what they found is that when they look at the data during sleep, and they look at the slow waves, so this shows an example slow wave or downstate, the up to down transitions were synchronous, but when they ordered the cells from dorsal to ventral, the dorsal cells basically always seem to spike earlier.
Иван Купала. Зона поздравляет.
And so basically what they have is this traveling wave, but it's a unique kind of traveling wave where it's not really the delta wave that's traveling, but the down to up transition, so the return to the up state travels. And so we're able to quantify this when we look at the up onset delay, so the delay of spiking as a function of depth. We see a nice correlation. We only see this when we look at the down to up transition, and not, at least on average, in the significant correlations in the up to down transition. And we can reproduce this when we look at the gamma power, so a good proxy of spiking aligned to the slow wave. You see that basically all the channels from, or a subset of channels from dorsal to ventral, tend to fall into the down state at around the same time, while the return to the up state starts with the dorsal channels and travels to the ventral channels. So how might our model be able to explain this? Basically, what we have in the model is a number of different parameters that might be able to shape the down to up transitions. And we can use that to ask, okay, do these dorsal channels behave as if they're more excitable? Do they behave as if they're more recurrent? What in our model might explain this traveling wave? And so what we're going to do is build a spatial gradient into the model with a variation in one of these parameters along the gradient and see which parameter can result in the observed dynamics. So we started with excitability based on some observations that cells with higher firing rate tend to fire earlier in the up state, thinking that, okay, maybe these cells are more excitable. And so this shows a simulation of the model now with a gradient in excitability from a dorsal to a ventral population. And when we align the activity in the model to the up to down and the down to up transition, we actually find that this excitability gradient isn't able to reproduce the data, right? So the more excitable or dorsal population not only goes up sooner and travels, so that does match the data, but it doesn't go from up to down synchronously. So it also stays up longer at the end of the up state. And so it actually turns out that none of the gradients in the model are able to produce the up-down asymmetry. In all cases, you get an up-to-down transition that isn't synchronous, which was kind of the really key feature that we saw in the data. And I've only showed this with a single parameter example, but when we do a sweep through parameter space, we see that this is the case. So at this point, we kind of took a step back and say, that's a little disheartening. What can we do to our model to capture this? And so I was awesome. Thanks. I'll go through this really quickly. Just to say that I was reminded of something I discussed with John during my PhD, which is that a hyperpolarization activated inward current might act as a functional adaptation. So this is a property by which when you hyperpolarize cells, they tend to depolarize. And so in the model, this is basically adding a double negative, right? So instead of having something that at high input inhibits the network, we have something that is inactive at high input, but at low rate activates the network. So these can effectively act as the same thing, even though one is an excitable or an excitatory current, one's inhibitory. And we can, with this rectifying current, also produce excitable up dynamics, just like we see in the model. And when we build in a gradient now of high IH dorsally and low IH ventrally, we're now able to reproduce what we see in the data. So we have a synchronous up to down state, but the down to up transition travels from the dorsal population to the ventral population. And this is basically because now we have a configuration in which the dorsal and ventral populations are affected the same during high rate, so during the up state, but during the down state, during low rate, the dorsal populations activate this H current stronger, so they return to the up state earlier. Okay, so conclusions. The interplay between recurrence and adaptation can give 4 different regimes of alternation dynamics, 3 of which are metastable, so we have a bistable and 2 excitable regime. The slow oscillation during non-REM sleep best matches one of these excitable dynamic regimes. So it has a metastable up state with a transient down state. And what we find when we look at the head direction cortex and the fact that we see this traveling up state onset suggests that actually the H current might be the responsible player for downstate transients rather than traditional adaptation currents. And so I just have to thank my PhD advisors, Yuri and John, Brendan, who collected the data for the first half of the talk, my postdoc advisors, Adrian and Blake, Adrian, who collected the data that I talked about in the second half of the talk, and Drew, who I've been working with on the analysis of that data. And thanks, Thilo, for organizing this symposium, and everyone for your attention.
I'm muted. So yeah, thank you very much for this beautiful talk. And yeah, we are already a little bit late, but maybe a very brief question, or one very brief question, if there is. Otherwise,
I think Mauricio has one.
All right, so Mauricio.
Very fast. Beautiful talk. I like it very much. Can you exclude the possibility that the synchronization is coming from outside the cortex?
No, you can't really. And yeah, I mean, I guess if you have your upstate is stable, and here, let's go back.
If it's a long answer, please, maybe you can also discuss after Luca's talk.
Yeah.
So
I don't think we can exclude that, but let's talk about what that would mean, because once you start thinking about the input, you know, things get complicated, as you know. Yeah.
okay.
Thank you.